until I graduated high school, and I've spent most of my life abroad studying and working. I love dogs and I enjoy good wine and food, but mostly I love to travel and meet new cultures. So quickly, let's go over to what Urania already said about me. My career started when I got into medical school back in 2001. Then I continued my education with a master's in environmental health. And while doing my PhD in the Netherlands at the University of Groningen, I also participated into more courses and received a postgraduate degree in epidemiology by the Netherlands Epidemiological Society. Then, before defending my thesis, I moved to London for a postdoctoral research project in a different area of epidemiology, mainly on systematic review and meta-analysis. For a short while, I relocated to Cyprus, and I was teaching epidemiology to medical students. But I always, the question about returning to clinical training was there. So in 2017, I decided to go back to clinical training and become a general practitioner. So in the past 4 years, I combined my clinical training with research at the University of Crete. And this brings us today, being a new member of the family of the University of Nicosia Medical School and a proud faculty member of the Department of Primary Care and Population Health. So for the past 10 years, my research, as Urania already mentioned, was mainly focused on allergic respiratory conditions. I started by investigating genetic and environmental factors affecting asthma severity and its underlying airway pathology in adults. I also worked with child populations and collaborated with European birth cohorts. My postdoctoral work was focused again on a broader area of allergies and comprised of systematic reviews relating to infant feeding and allergy and autoimmune disease development. More recently, I've been collaborating with the rare birth cohort in Crete, investigating early life risk factors and child health. And I've also been doing research on primary care with real-life clinical data from COPD patients. I have investigated clinical, behavioral, and treatment characteristics of Greek COPD populations in primary care. And I have also supported clinical research within different other disciplines with colleagues from the university hospital. So I started my career with a doctoral thesis on genetic and environmental interplay in asthma severity within the Groningen Research Institute for Asthma and COPD. The most important part of that asthma study was that on top of clinical follow-up assessments, it had genetic and also histological data available to better understand the underlying mechanism of airway pathology. Part of the thesis was completed at Harvard University, where I focused on air pollution exposure and gene-air pollution interactions on asthma outcomes in children. Asthma is triggered by different environmental stimuli, including allergens, airborne pollutants, viruses, weather changes, and exercises. It involves bronchoconstriction, excess mucus production, local inflammation, and edema in the airway wall, leading to airflow limitations, which usually are reversible. However, structural changes of the airway will also occur and can lead to fixed airflow obstruction. Airway wall comprises of pathological changes in the airway, including disrupted and denuded epithelium inside of the airway wall, basal membrane thickening, subepithelial vessel hyperplasia, mucus glands hyperplasia, as well as hypertrophy of airway smooth muscle wall. This can lead to narrowing of the airway lumen, and which will cause eventually airflow limitation and symptoms in asthmatic population. The mainstay hypothesis of my thesis was that asthma is an epithelial disease. We focus on the fact that the airway epithelium of asthmatics is more susceptible to damage and has altered responses to injury by environmental stimuli. This injury and impaired repair will lead to interaction between the affected airway epithelial cells and the underlying mesenchymal and immune cells with the release of cytokines and growth factors, leading to inflammation, local sensitization to environmental allergens, and chronic and persistent airway remodeling. Eventually, all this will lead to clinical severity with lung function limitation and lung function decline over time. Nevertheless, there's heterogeneity in clinical and pathological expression of asthma. That means that not all asthmatics respond to environmental exposures in the same way, neither they all have the same severity in clinical expression. The individual variation in asthma severity is influenced by interactions of genetic and environmental factors. The primary aim of my thesis was to investigate the role of genes encoding proteins involved in epithelial barrier integrity, chronic airway inflammation, and remodeling. We used candidate gene approach to test for associations of single nucleotide polymorphism and pathological outcomes seen in bronchial biopsies and clinical severity outcomes such as lung function level decline and air hyperresponsiveness, common features of asthmatics. We also aim to assess whether the genetic effect was modified by inhaled corticosteroids, the mainstay therapy for asthma, or smoking in asthmatics. We used Dutch asthmatic populations. All subjects were adults and atopic. Population 1 comprised of selected patients from past cohorts and were invited for a cross-sectional clinical evaluation and bronchial biopsy collection via bronchoscopy. The second population was a prospective cohort study of asthmatics with multiple lung function measurements, recorded inhaled corticosteroid use over a period of 25 to 30 years. There was an overlap between the two populations. The rationale for selecting our candidate genes were the following. We selected EC-adhering as it is one of the main adhesion molecules able to establish and stabilize cellular junctions and maintain epithelial integrity of the airway. Inhaled corticosteroids have been shown to upregulate the protein in epithelial cells and restore epithelial integrity. And it has been related to airway wall remodeling as well. Urokinase plasminogen activator receptor, it's a protein implicated in epithelial cell proliferation, and its gene had been identified as asthma susceptibility gene by other colleagues of my research group. And also variants on that gene were associated with lung function decline in asthma, as well as airway hyperresponsiveness. Finally, TGF-beta1 is a cytokine that is increased in asthma and has been related to airway remodeling. and variants on that gene had also been associated with asthma development and severity, while smoking has been shown to increase this cytokine expression and has also been involved to more severe asthma expression. This work has concluded in 3 publications, one for each candidate gene study. And the main conclusions were that indeed there is genetic susceptibility in hearing expression and epithelial integrity. Also, epithelial cell proliferation has a genetic background, as well as the airway wall remodeling features and airway inflammation. Importantly, this genetically determined epithelial integrity and remodeling contributes to clinical severity in asthma with accelerated lung function and decline. We show consistent direction of association for variants between clinical and pathological outcomes. And this consistency supported the hypothesis that there's genetic variability starting from airway epithelium and leading to clinical severity in asthma. Moreover, we've seen that some of these effects were modified by inhaled corticosteroids or smoking. And importantly, we confirm a beneficial effect of inhaled corticosteroids in these asthmatic populations. The secondary aim of the thesis was to determine whether lung function susceptibility is influenced by, whether a lung function susceptibility of asthmatic children to ambient air pollution. We also wanted to assess pollution effect modification by inhaled anti-inflammatory medication and genetic variation, and this time on a genome-wide scale. For answering this research question, we used data from a multi-center randomized clinical trials in 8 North American cities, which included asthmatic children of mild to moderate asthma severity. The primary aim of the CAMP study was to assess the long-term effects of daily inhaled anti-inflammatory medication, including inhaled corticosteroids and metoclopramide. We had data from a 4-year follow-up with visits at every month, for which we had spirometry. And then retrospectively, we collected 24 averages of gaseous pollutant exposures from the EPA for ozone, carbon monoxide, nitrogen dioxide, and sulfur dioxide, and estimated exposures for the same day, 1 week, and 4 months moving averages prior to each spirometry of the children. We saw that air pollution adversely influences lung function in asthmatic children. On the left figure, we can see that for both FEV1 on top and FVC at the bottom, carbon monoxide for each level of exposure, same day, 1 week, or 4 weeks or 4 months moving averages, was associated with decrease in FEV1 and FVC. We've seen this adverse effect also for the 4-month moving averages of nitrogen dioxide. However, while we assess whether regular treatment with controller medication had a mitigation effect against the pollution adverse effects on lung function, unfortunately, we didn't see what we expected. You can see on the right-hand side figure, either for FEV1 on the top panel or for FVC on the bottom, that for placebo, budesonide and nedochromate group, we don't see significant differences on pollution effect. Nevertheless, this clinical trial design gave us the opportunity to assess modification of pollution effects by treatment without confounding by indication. And what that means that we can be more confident about the relationship we see, meaning that we exclude the fact that these children will be exposed to higher levels of exposure and then use more medication, and the effects we'll see will be biased by that effect. These were randomized children, and so we don't expect to see confounding by indication. After we published the main work on lung function susceptibility from air pollution effects, we wanted to test whether these genetic variation, these effects were modified by genetic variations. We used a two-step method for fast linear mixed model computations for genome-wide association study. And we also used another method called improved gene set enrichment analysis for GWAS to identify plausible pathways involved. We concluded that both the SNP at the SNP level and at the pathway level analysis, that the host lung function response to long-term exposure to pollution is linked to genes involved cellular adhesion, oxidative stress, inflammation, and metabolic pathways. We were happy to find pathways related to cellular adhesion and epithelial integrity, which again supported the original hypothesis that indeed the airway epithelium may play a role to responses to environmental stimuli. Outside of the scope of the thesis, we collaborated with other European birth cohorts and North American cohorts, and we perform another genome-wide interaction study followed by functional genomics analysis, and indicated again the involvement of several genes at the genomic, epigenomic, and transcriptomic levels for asthma-related air pollution exposure. These results support the notion that gene-environment interactions are not important only for asthma severity, but also for asthma development in childhood. So moving on to the next level of my career and to evidence-based medicine. My postdoctoral work was mainly focused on the concept of developmental origins of health and disease hypotheses, which links a range of perinatal exposures, including nutritional exposures, to virus and specific sensitive periods of development, to lifelong health consequences. The areas of interest of these early life exposures of this project were mainly the infant feeding at early life stages, including dietary intake and supplements during pregnancy and lactation, duration of breastfeeding, and special formulas used for high-risk infants, allergenic food avoidance, and timing of solid food introduction, and more specifically, timing of allergenic food introduction. This systematic review was supported by the Food Standard Agency of UK, and the outcomes of interest were all atopic and autoimmune diseases. It was a huge systematic review, which resulted in different smaller systematic reviews. We started with 16,000 titles to be screened, and we ended up with over 1,000 relevant articles for data extraction and analysis. At this stage, let me go back and remind you that until recently, we delayed giving allergenic foods to infants for a previous hypothesis that the delay of allergenic food introduction may actually reduce the risk of food allergy. However, for the past 20 years, the prevalence of food allergy has been on the rise worldwide. What this new study provided to the overall evidence was actually that the introduction of egg and peanut at early age, as early as 4 to 6 months, may prevent the development of egg and peanut allergy, the two most common food allergies around the world. However, we should be cautious with babies who already has a food allergy or eczema, and of course, these results should not interrupt the exclusive breastfeeding for up to 6 months. There's no reason to stop exclusive breastfeeding for early introduction of these foods. In addition, this meta-analysis showed no evidence that early introduction to gluten increases the risk of celiac disease, and this evidence was graded as high. Overall, these results led to change in recommendations for infant feeding in allergy prevention. In addition, we found evidence that probiotics during late pregnancy and lactation may reduce risk of eczema. Similarly, fish oil supplementation during pregnancy and lactation may reduce risk of allergic sensitization to egg. Weaker evidence was found to support that breastfeeding promotion reduces risk of eczema, and longer exclusive breastfeeding reduces risk for type 1 diabetes mellitus. Also, weak evidence was found for probiotics and reduction in risk of allergic sensitization to cow's milk. We did not find any evidence that other dietary exposures, including prebiotic supplementation, maternal allergenic food avoidance, vitamin, mineral intake or supplements, fruit and vegetable intakes influences risk of allergic or autoimmune diseases. Last but not least, we wanted to assess the previous recommendations that high risk of allergy should be initiated with hydrolyzed formulas instead of standard cow's milk formulas. What we showed, it was that overall there was no consistent evidence that exclusive or partially hydrolyzed formulas reduce risk of allergenic or existing risk of these outcomes. This is shown in this forest plot, which shows the pool estimates of the individual meta-analysis for each outcome. On the left, for exclusively hydrolyzed formula, and on the right for partially hydrolyzed formula, and you see that the overall pool relative risk is not significant. Again, this work influenced infant feeding guidance in UK, but also internationally. Allergy, I thought would be relevant and interesting to also discuss about food-related anaphylaxis reaction and admissions. And this again was work done in collaboration with my colleagues at Pediatric Allergy of Imperial College. And what we did, we wanted to see time trends in anaphylaxis in England and Wales from 1992 to 2012. And we've shown that there was an increase in anaphylaxis-related hospital admissions, but luckily no increase in fatalities. More recently, we've done a similar work with data up to 2018, including the whole United Kingdom. And again, you can see that there's an increase in food-related and not food-related anaphylaxis reactions in both males and females. I want you to see the bottom panel of figures and see that drug-related anaphylactic reactions usually occur in senior adults, while food-related reactions usually occur in children and early and young individuals, young adults. Again, we show that case fatality rate was not increased, but on the contrary, actually the case fatality rate decreased over the years, suggesting a better education for both the patient with food allergy or known allergies in general and the healthcare professional, and better care for these patients. So more recently, I still keep my interest for research in allergy, and I collaborate with local research groups as well as international groups of primary care and allergic rhinitis and asthma. But I'm also interested in prenatal exposures and child health. And there, I'm going to show you some work being done with the data from the rare birth cohort in Crete, and also some work on primary care research for COPD patients. The rare birth cohort was a mother-child prospective cohort set up in Crete around 2008. And the main objective was to assess different environmental exposures early in life and what those exposure, how this exposure may influence health later on. Recently, we have completed the 10-year follow-up of children born from this cohort. And we have information on different exposures, including air pollution exposure, persistent organic pollutants, heavy metal toxicity, and diet for mother and children. And collection of on information of outcomes, including allergic, cardiometabolic, and neurodevelopmental outcomes, was mainly collected through questionnaires during follow-ups. But also there's a rich bank of biosamples, including cord blood, urine, and serum samples. Omics data are also available in the rare cohort, which participated in many other European consortium of birth cohorts. As you see here, the burden of allergies also in Crete is relatively significant, with children reporting ever doctor diagnosed asthma up to 12%, eczema, ever having eczema up to 23%, and rhinitis symptoms in the last 12 months, again, up to 23%. Seeing this prevalent allergy in the Ierodiakonou birth cohort, we wanted to assess whether prenatal air pollution exposure may be one of the reasons of allergy development. Together with the help of a postgraduate student of the Masters of Public Health of the University of Crete, we tested this association and found that higher prenatal particulate matter exposure increased the risk for eczema and allergic rhinitis in childhood. We did not find associations with asthma development, as previously shown, but a recent systematic review also indicated that there's no consistent evidence that prenatal exposure to traffic-related pollution is associated with risk of asthma. We also assessed effect modification by maternal allergy and maternal adherence to Mediterranean diet during pregnancy. And we show that pollution effects were more pronounced in children with a history of maternal allergy, but unfortunately, effects of pollution were not mitigated by adherence of the mothers to the Mediterranean diet during pregnancy. Other exposures of interest were prenatal exposure to cadmium. Cadmium is a heavy metal to which we are in general exposed through diet, through food, and it has been shown to play a role in bone health, to influence bone health. And what we've seen is that elevated prenatal cadmium levels in maternal urine during pregnancy were significantly associated with slower trajectory of weight and height gain in the children of this cohort. And some of these effects were more pronounced in girls and in children born to mothers who smoke during pregnancy. Smoking is another source of exposure to cadmium. Moreover, we wanted to assess whether the recent work being done on sleep deprivation and cardiovascular and cardiometabolic disease seen in children and adults can be confirmed for gestational sleep deprivation and effects on child cardiometabolic outcomes. We concluded that gestational sleep deprivation, defined as the pregnant women sleeping less than 6 hours per day, was associated with higher BMI in children and risk for overweight and obesity, as well as with higher diastolic blood pressure in childhood. The observed associations, again, were more pronounced in girls and partly mediated by gestational diabetes and preterm birth. Before closing up, I would like also to describe the work being done with COPD patients from primary care in Greece. The Greek Unlock study was a cross-sectional study exploring outcomes in COPD patients. And here I provide the main conclusions of that work which has been published in several articles, showing that the majority of COPD patients reported uncontrolled symptoms and poor adherence to inhalers. Worse health status and COPD severity are associated with the poor adherence reported in these patients, but also worse disease is shown in subjects with poor sleep, with frailty, and low self-efficacy and sense of coherence. Frailty with mobility disability and multimorbidity are prevalent in this group of patients. And surprisingly, we found that healthcare professionals do not strictly comply with treatment recommendations for these patients, and that was obvious by the overprescription of inhaled corticosteroids in COPD patients. So what is up for the future? I had submitted a proposal for the GSK supported study program and received a grant to study severe asthma, nasal polyposis, and biologics in these patients in collaboration with the Department of Respiratory Medicine of the Heraclion University Hospital. Again, the main hypothesis of this proposal is the epithelial barrier integrity and its role in severe eosinophilic asthma with nasal polyposis. The study aims to include 3 phenotypes of asthma: severe asthma with polyposis, severe asthma without polyposis, and polyposis with mild or no asthma. And the objectives are to assess and compare airway remodeling in the 3 different phenotypes, response to biologic therapy between and within phenotypes with focus on airway remodeling outcomes. And finally, the main objective of the study is to assess whether shedding of epithelial barrier cells and proteins, including ICA hearing, in respiratory secretions of patients with severe asthma could become a useful novel biomarker. There's a long list of people that I met and supported my studies and my research in the past years that I would always like to thank, as well as my some research groups and societies. And of course, before closing this presentation, I would like to invite any one of you who shares similar interest for research to contact me and discuss ideas for future research under the umbrella of the University of Nicosia. Thank you all for your attention. And I would like to take your questions now.
Okay, Despo, thank you very much. This was a very comprehensive summary of your very interesting research work and also very impressive research work. And I think because we have a lot of students who are here today, I think we had about 50 people at some point attending, and a lot of them are students. I think I want to point out to them, especially the systematic review on the nutrition from pregnancy to early years of life in relation to the development of atopic and autoimmune diseases, and kind of tell them that this is an example of how evidence-based, how research can be incorporated into evidence-based medicine and change recommendations and practice, which is, which is an outcome that I think all researchers would like to come to at some point in their research work. So well done for doing it so early in your research career, because
Thank you.
It has changed the recommendations, and now we know that we can expose our children to allergens very early in life, because in the past we thought it was different. Nobody has written any questions. We have a comment from Avis. And his comment is on your study on the hydrolyzed formulas, show that it doesn't change the risk. in terms of developing allergic diseases in children. And he said it was a shame because he had to feed his baby this disgusting hydrolyzed formula
and pay a lot of money.
Pay a lot of money and not do anything about it. Yes, okay, go ahead.
Sure. I just wanted to mention that, I was lucky and happy to be involved in such a wonderful project with, indeed, after all the hard work, had clinical implications. And as you said, it is important for students to understand that this is why research is being done, and because what we do, we accept it as correct, but over time we can see that indeed recommendations or guidelines can be changed based on research and evidence.
Mm-hmm. I'll go ahead with one question that I have. You did say, again, as part of that systematic review, you did say that exclusive breastfeeding was, there was some evidence that, that breastfeeding promotion or exclusive breastfeeding, I can't remember which one of the two it was, that it kind of reduces the risk of eczema, but not other allergies from what I understand. Did you look at all whether this association is different in children who have genetic predisposition to allergies or not?
With respect to genetic, well, most of these observational data was focused on diet and allergic outcomes. However, many of them, they separated children with family history and not. So we did this subgroup analysis and we didn't see different effects. And basically that was the only indication for genetic susceptibility and whether that could modify the associations.
Okay, that's interesting because I think in the case of cesarean section and association with allergies, still the evidence is not, I don't think there's hard evidence, yes, but I think there was some kind of evidence that it might be might be different, this association in children.
Yeah, we show that with the prenatal exposure to air pollution and association with eczema and rhinitis. Children from mothers with a history of allergy had higher risk compared to children of mothers with no previous reported allergies.
Any questions from the audience? Come on, guys. Any more questions from your end? I mean, you do have a lot of, Despo is new to the team, but she has a lot of common interest with the rest of the epidemiology group. And although she has been hired more towards the primary care team, we will, of course, invite her to participate in our epidemiology project because we have a lot of common interests. And she has experience from the birth cohort study in Crete as well, which is, again, one of the areas that we would like to try and see whether we can set up something similar inside.
Just since you mentioned the birth cohort, colleagues there are really interested in continuing collaborations and share data for new research hypotheses. So anyone who might be interested into working with the rich data or information from the REA, happy to bring you into communication with Lida Hadzi and Marina Vafiadi and work on research questions.
Yes. Any other?
I see a raised hand by Abius. Is that for a question or is that?
Can you? Because let me just see. Yes, Abius has. Let me just see if I'm not sure I can unmute him because I haven't got host. Abius, can you write your comment in the chat box? Yorgo, can you unmute Abius? And we have somebody else who has raised hand as well.
Let me just see if I can do that.
Yes, thanks. It's interesting, the sleep study. Actually, I read the study as well, and I was very much interested in that. But you said that it was mediated, the effect was mediated by gestational diabetes and birth weight.
And preterm birth. Yes, I can elaborate more on that.
Can you elaborate a little bit more on that?
Yes, so the reason we wanted to see if there was a mediating effect by perinatal factors was that there have been studies showing that sleep deprivation during pregnancy is associated with adverse perinatal outcomes, including preterm birth, gestational diabetes, preeclampsia, and so on. So we saw a relationship between gestational sleep deprivation and cardiometabolic outcomes in children. So I wanted to see whether this was mediated by these factors. And indeed, the mediation analysis we've done, and that was again in collaboration with a group, the ABCD cohort study. Actually, these findings were replicated in a Dutch cohort, which I forgot to mention, or I wanted to make it shorter. So indeed, we show that basically sleep deprivation during pregnancy leads to gestational diabetes, and part of the effect we see in children is because of gestational diabetes. Similarly for preterm births, yeah.
So the other, I think, okay, so So the mother did not during pregnancy and didn't get gestational diabetes shouldn't blame themselves if their children are obese.
Well, it was partly mediated.
Okay, so there is still some.
There are still some other factors that may mediate the effect, or maybe we still show the direct effect, but we saw also a part that it was mediated. Part of the effect was mediated by these perinatal factors.
Okay, we have a question, Christian, but I can hear my voice back, guys.
Okay.
Basically, I think.
I will unmute Avis now for his question, and then a student. Avis, go ahead.
Hi, Despo. Yeah, thank you for the talk. It was really interesting.
Thank you, Avis.
I'm a neuroscientist, so I don't understand half of the stuff there, but it's quite intriguing about sleep. So from what I understand now, sleep deprivation leads to maternal diabetes, and that leads to the newborn complications, right?
Yes, that's what we show, that the effect is partly mediated by the gestational diabetes. There's still direct effect, and we basically we quantify these in the analysis. But yes, it is true that sleep deprivation can lead to gestational diabetes, and gestational diabetes then is associated with the childhood obesity.
I think the interesting point could be to distinguish between paradoxical sleep and non-paradoxical sleep, so slow wave sleep. If this is the...
Unfortunately, yeah, that was the problem, and one of the limitations of the study. We didn't have information on sleep quality. We only had about sleep quantity for REA. So we couldn't really assess, besides the quantity, whether different types of sleep quality or could play a role. And that was a study, so it was questionnaire based.
You can probably use those watches now that tell you in which stage of sleep you are,
Yeah, that's a good idea for a new study. Yeah, I have one. Actually, it says my sleep quality. Yeah.
Okay, thank you.
Do you think the sleep quality provided by this smartwatch is reliable? The information we receive back?
Yeah, I think you have to get an EEG, but obviously that's a bit more challenging for the subject. But
yeah,
it's not. I don't think it's too bad.
Mm-hmm. Mm-hmm.
Yeah.
Good.
Okay.
You gave me a new idea now.
Okay. And one question from Liran Bitton.
And we have a comment from Christian as well that I will read after the question from the student.
Yes, hi, I'm a medical student, and I'm interested in research. And I wanted to know upon what basis you decide to begin research in a specific field or area of study. Like, what drives you to say, I want to begin doing research on this?
Okay, so that's a good question. So originally, when I was young, around your age, and I was completing then my medical qualification, my primary aim was to become a respiratory physician. And that brought me into the research of asthma when I decided to do research. But over time, in being more involved in epidemiology and broader areas of interest and research, I really understood that I really also like to have real world data and being closer to that and information coming from primary care. And so I ended up becoming a GP instead of respiratory physicians, but with special interest again in asthma and respiratory diseases. So it's something you begin with, but it doesn't have to end with your primary aim. That's the beauty of research. You gain skills over time and more interest, and you can switch areas of interest. There's no limitation. Sky's the limit. So you don't have to think too much about, I have to find the area that I want to study forever, but instead, do something that at the moment you like, because of the research question, because of the subject, because of the supervisor, and then over time you find your way. I don't know if Urania or other senior staff agree with what I just said.
I mean, that's very true. As Despo said, you just need to develop your research skills, and obviously, you would be able to decide later on whether this is a field that you want to do research in or whether you want to move into a different area. But depending on the opportunities, of course, as well. But that's true. I mean, a lot of the skills are transferable from one area to another.
Exactly.
True. Okay, we only have a couple of minutes left. There is a question from a student, Christian, who says that corticosteroids with long-acting beta agonists have been shown to lead to better quality of life and fewer asthmatic attacks when compared to steroids alone. Will your findings be true with the approach of using both medications since you studied only steroids alone?
That's a good point. So remember that this was a study done in early 90s, 93 to 98, mild to moderate asthmatics. So I believe at the time, as it is for now, for mild asthmatics, monotherapy with inhaled corticosteroids is suggested for mild ICS LABA also as needed. So the trial at the time was done for steroids and metoclopramide, which was another anti-inflammatory inhaled medication used back then. But obviously there are a lot of studies showing indeed the benefit of the combination therapy for asthmatics. And the research question of my study was not about the effectiveness of the inhaled medication, but whether pollution, but whether the medication modified the effect of pollution. So it's a different question.
Thank you very much, Despo. I think we've reached 6 o'clock, so we need to close the meeting. So first of all, I would like to thank Despo for her excellent presentation today. And again, welcome here to our department and our medical school. And we're looking forward, of course, to collaborating with her in all areas of research and not only research, but all the other pillars of our department. I would like to thank everybody for attending today. As I said, we had almost 50 people attending, which was very good, and everybody for their questions. And just to say at the end that we will not be providing the slides of the presentation to the students that were asking. This is not part of your curriculum. This is an introductory lecture by new faculty members, so you're not being tested, you're not being provided with the slides, but we are hoping that we will have a YouTube channel to upload the presentation at some point, so you might be able to find it there. So thank you again. I think we had a few comments again at the end. I think from a few students and faculty members saying that your presentation was excellent, Despo. They enjoyed it. Looking forward to working with you.
Thank you very much. It was a pleasure for communicating my work and experience, also with younger students, medical students, and also invite any student who might be interested in doing research to contact me and discuss ideas for their projects or for research in general. Thank you all. Thank you, Rania.
Thank you.
Happy to be part of your team.
Thank you, and thank you, Yorgos, for organizing this. So have a good evening, everybody. Bye.
Bye. Thank you all. Bye.